Determination of the dissolved oxygen content of water. The objective of this experiment is to determine the dissolved oxygen content in the given water sample. The apparatus needed in this experiment are iron stand, burette clamp, acid burette, BOD bottles, Erlenmeyer flasks, pipettes of different sizes, pipettor, and graduated cylinder. The materials needed in this experiment are manganese sulfate solution, starch solution, concentrated sulfuric acid solution, alkaline iodide azide solution, and sodium thiosulfate solution. Dissolved oxygen determination measures the amount of dissolved or free oxygen present in a volume of water or wastewater. Dissolved oxygen is used as an indicator of the health of a water body where higher dissolved oxygen concentrations are correlated with high productivity and little pollution. It is a measure of the ability of water to sustain aquatic life. Aerobic bacteria and aquatic life, such as fish, need dissolved oxygen to survive. If more oxygen is consumed than is produced, dissolved oxygen levels decline, and some sensitive animals may move away, weaken, or die. Dissolved oxygen is determined by the titrometric method developed by Olaus Winkler, called the Winkler method. This technique is used to measure dissolved oxygen in freshwater systems. This test is performed on-site as delays between sample collection and testing may result in an alteration in oxygen content. The preparation of the sample. Fill the 300 ml BOD bottles with a water sample up to the brim. When full, cover the bottle and remove excess water. Make sure that no bubbles are formed. Immediately add 2 milliliters of the manganese sulfate solution to the collection bottle by inserting a calibrated pipette just below the surface of the water sample in each BOD bottle. Squeeze the pipette slowly so no bubbles are introduced via the pipette. If the reagent is added above the sample surface, you will introduce oxygen into the sample. Add 2 milliliters of the alkaline iodide azide reagent in the same manner in each BOD bottle. Stopper the BOD bottle with care to make sure that no air is introduced. Mix the sample by inverting several times. Check for air bubbles. If oxygen is present, a brownish-orange cloud of precipitate or flock will appear. Add 2 milliliters of concentrated sulfuric acid solution using a pipette held just above the surface of the sample. Carefully insert the stopper and invert several times to dissolve the flock. When the flock is dissolved, the sample is considered to be fixed. The titration process. Measure 203 milliliters of the prepared sample in the BOD bottle. Transfer this to a 500 milliliter Erlenmeyer flask. Take the initial reading of the volume of sodium thiosulfate solution inside the burette, which has been previously rinsed with the same solution. Add 2 milliliters of the starch solution into the sample in the Erlenmeyer flask. Make sure to take note of the changes in the color of the sample. Titrate the sample slowly with 0.025 normal sodium thiosulfate solution until the endpoint is reached. The endpoint is reached when the blue color of the sample disappears from the last drop of the titrant. During titration, be especially careful that each drop is fully mixed into the sample before adding the next drop. After adding each drop of the titrant, swirl the Erlenmeyer flask containing the sample. Take a final reading of the volume of the sodium thiosulfate solution in the burette. The data for the other two trials will be given by your instructor.